Hello, everyone, and welcome back. In this lecture, we will cover the biological macromolecules. The early formation of planet Earth is referred to as the Hadean period, from the Greek god of the underworld, Hades. It was an incredibly inhospitable world newly formed. The planet was incredibly hot while still in the process of clearing its orbit and being bombarded by asteroids and other objects from outer space. While the temperature was well above that for which complex life known today could survive, scientists still want to understand how the process of life progressed on Earth. During this time period, the atmosphere consisted of mostly carbon dioxide, methane, and ammonia, unlike our atmosphere today, which is mostly consists of nitrogen and oxygen. Stanley Miller and Harold Urey started a series of experiments in the 1950s. The goal was to simulate hydrological cycles like on Earth. In the center of the image to the right, you can see that they added water, methane, ammonia, and hydrogen to a closed loop system. At the bottom left, you'll see liquid water that is heated into water vapor, then cycled through and cooled again into liquid water. Additionally, spark was introduced to simulate lightning. The hypothesis was that by stimulating early Earth environmental conditions coupled with lightning, the synthesis of some of the complex biomolecules that we know are the basic ingredients of life would occur. If this were not corroborated by the evidence, then the alternative hypothesis, called the null hypothesis, will be that after the experimentation, only the starting molecules will be found in the liquid water. And so, what are the basic ingredients of life? I find many of these macromolecules important to our own nutrition. The carbohydrates are things like simple sugars, or our more complex sugars called starches. Lipids, which we signify on the nutritional label as fats. Proteins, which serve as enzymes for our body. And finally, nucleic acids, which serve in large part as a storage of information in cells. There are four distinct macromolecules that all life as we know it have. In the previous chapter, we examined molecules with about two atoms. However, macromolecules are made up of many atoms, shown as circles in the depictions. These macromolecules appear to be no easy task of forming because cells go through great lengths to synthesize them. These molecules are what Miller and Urey set out to synthesize without the mechanisms of a complex cellular machinery. Mono is from the Greek word for one, and poly from the Greek word for many. A monomer is a single molecule that can be combined with similar molecules to create larger structures. Most macromolecules are polymers, and polymers are made up of many monomers that are bonded together. Our bodies take these macromolecular monomers from the food we eat and builds polymers out of them to make the different components of our cells. To do so, it uses what's called dehydration synthesis reactions. By examining the words, can you make an educated guess as to what dehydration synthesis reactions would mean in this context? Dehydration synthesis is when two parts, in this case two monomers, are synthesized together and one water molecule is formed and released. In other words, dehydrated in the process. Suffix lysis means to break down. Therefore, hydrolysis or hydrolysis reactions are the breaking apart of molecules with the addition of water molecules. To take a look at how each of the four classes of macromolecules are formed, we'll need to know more about their subcomponents. A functional group is a substituent of a macromolecule that gives the molecule a special function in the way the chemical reacts with other chemicals. These functional groups are found in the four classes of macromolecules and will come up over and over again in any biology class you take. They involve hydrogen, carbon, nitrogen, and phosphorus. The dashes between atoms represent a covalent bond, which remember is an electron from each atom being shared. We've already discussed phosphates found in the head of a lipid, but there is also the hydroxyl group, which is an oxygen with a covalent bond to a hydrogen. A carboxylic acid group, which is a carbon with two bonds, also called a double bond to one oxygen, and a single bond to a hydroxyl group. Phosphate, which has one phosphorus atom surrounded by oxygens and a hydroxyl group. Lastly is the amino group, with a nitrogen bonded to two hydrogens. From here, we can take a closer examination of the macromolecules, beginning with carbohydrates. Carbohydrates, meaning carbon water, are rings of carbon atoms bonded to hydroxyl groups. The first molecule listed is glucose. It has 6 carbons, 12 hydrogens, and 6 oxygens. More elegantly put, it has 1 carbon for every H2O. There are 3 very common sugars, but you will find there's an enormous array of them. Glucose is the sugar used in our bloodstreams and from which other sugars are converted by our bodies. Galactose is found in milk, and fructose in fruit. All sugars end in the suffix ose or ose. Let's take a monomer of glucose and see how it forms a polymer. As we see here, we have a hydroxyl group bound to one sugar and another hydroxyl group bound to the other sugar. And you can see the red boxes highlight a hydroxyl group and a hydrogen on one of the hydroxyl groups. And you may have guessed, we can do a dehydration synthesis here. The H2O molecule bonds are broken and reformed to give off water. And the carbon now is available to react with the other sugar's oxygen. And here we have a newly formed polymer. In this case, we went from 2 monosaccharides, mono meaning one, to 1 disaccharide. Di means 2. And this can be done over and over again to create longer chains of sugars into carbohydrates. Here we'll discuss the biological role of carbohydrates. Carbohydrates can be used for structure, such as the cellulose and plant cell walls, which are made up of entirely carbohydrates. And they can also be used as an energy storage system, such as glycogen, which is made up of entirely glucose molecules in the human liver. Checkpoint 1. How many water molecules would be generated from the dehydration reactions required to create a carbohydrate chain made of 5 monomers? Hint: you may want to sketch 5 sugars together and count the number of bonds formed between them from the dehydration synthesis reaction. Okay, now let's take a look at lipids, also sometimes called triacylglycerols. Let's break it down into its constituent components first. So lipids are going to be composed of multiple fatty acids linked to a glycerol. Glycerols are 3 carbon molecules with each carbon having 1 hydroxyl functional group. Fatty acids are long hydrocarbons or carbon and hydrogen chains with a carboxylic acid at the end. The fatty acid pictured here has 12 carbons in its chain and is called lauric acid. It is commonly found in coconut oil. Another very common fatty acid chain you may have heard before is palmitic acid and is found in palm oil. Checkpoint 2. What functional group is highlighted in the fatty acid molecule? Okay, now let's take a look at how the synthesis occurs. A common lipid is a triglyceride because it has three fatty acids attached to a glycerol. And we have two hydroxyl groups, and we're going to highlight a hydrogen from the hydroxyl group of the glycerol and the hydroxyl group of the carbonic acid group of the fatty acid. And these two can react with one another for a dehydration synthesis reaction, giving off water and forming a new bond between the leftover oxygen and carbon. And here we have a triglyceride, often denoted as a picture like so. Okay, now a special variety of a lipid that we need to be quite familiar with are phospholipids. Phospholipids, again, they have a glycerol. They have two fatty acids, but to the third carbon in the glycerol backbone, instead of phosphate, is bound. Sometimes this phosphate group has other atoms connected to it, but the key here is that there's a phosphate group, that P with four oxygens around it, bound to that final carbon in that glycerol. And this makes a phospholipid. We're going to review the biological role of lipids here. So, lipids, as we're all quite familiar with in today's society, uh, lipids are used for long term energy storage in the form of fat. Here we can see fat being stored around the body. But very importantly, lipids, especially phospholipids here, they're the main structural unit that composes cell membranes. So every one of your cells has to be enveloped by a membrane, and that membrane is made up of phospholipids. We discussed these phospholipids a little bit in the past here, and the polar nature of that phosphate head is going to be attracted to water, and those large hydrocarbons, which are a nonpolar covalent bond, they're not going to want to interact with the water. So they will automatically face inward. And because there's two layers, we have the polar phosphate heads facing the water on both the inside of the cell, which is a aqueous solution, as well as outside of the cell. So it makes this nice, neat little mosaic of phospholipids. Point 3. How many water molecules would be generated from the dehydration reactions required to create two triglycerides? And a note about saturated versus unsaturated fats. You've probably heard of these when we're talking about nutrition. Now, saturated fats, what that means is that the hydrocarbon tail, each one of those carbons is bound to as many hydrogens as it possibly can. Okay, carbon can form four bonds. And so, of those four bonds, each one of those carbons is maxed out for the number of hydrogens it can bind to. So, that's what we mean by saturated, is it's saturated by hydrogens. An unsaturated fatty acid, well, instead of having a complete saturated hydrogen bonding, some of these carbons, instead, they decide to form a double bond or share two pairs of electrons with their neighboring carbon. And this creates an unsaturated fatty acid. And here we can see two examples of this. Here we have butter, which is a saturated fat. And saturated fats tend to be solid at room temperature. And then we have unsaturated fats. A lot of plant oils are unsaturated fats, and they tend to be liquid at room temperature. Next up are the nucleic acids. You might be familiar with DNA as a very common nucleic acid. And acids, they're made up of monomers called nucleotides. Now, a nucleotide has three components here. The very bottom of that image, you can see the sugar, and the sugar is bound to two other very important components. Okay, one of them is a phosphate group, the other is a nitrogenous base. And it's called a nitrogenous base, well, because as you can see, there are several nitrogens in that compound. Okay. Oh, and importantly here, um, we can name the carbons that go around that sugar, and it tells us something very important, and it tells us which carbon the phosphate is bound to, which carbon the nitrogenous base is bound to, and something else that's very important is which number of that carbon is that hydroxyl group bound to on the sugar. So we can see the first carbon, called 1' has the nitrogenous base to it. The third carbon, 3' has the hydroxyl group bound to it. And the fifth carbon of that sugar has the phosphate bound to it. And that'll be very important when we look at how these nucleotides polymerize to form DNA. Okay, so we have our nucleic acids, and there are different nitrogenous bases that make up different nucleotides. The first three we'll cover are cytosine, uracil, and thymine. And these nitrogenous bases are made of a single ringed structure. And cytosine is present in both DNA and RNA. We'll cover those more in a bit. While uracil is only present in RNA, it's one of the defining features of RNA. And thymine is only present in DNA, a defining feature of DNA. There are two other nitrogenous bases, and that is guanine and adenine. Now, they are both present in RNA and DNA, and they have two rings. So the single rings are called pyrimidines, and the two ringed nitrogenous bases are called purines. Okay, now what is DNA and what is RNA? Well, an essential difference there is the ribose. So remember, ribose is the sugar group found in nucleic acids. And for ribose in RNA, if you look at the 2' carbon, it still has a fully formed hydroxyl group. In DNA, it has a 2' carbon where the oxygen has been removed, and that's called a deoxyribose. And hence the name ribonucleic acid for RNA, and DNA stands for deoxyribose nucleic acids. And these are the two main polymers of nucleic acids. And again, as discussed earlier, one of the defining features of DNA is it has thymine, but it does not have uracil. So it has guanine, adenine, cytosine, and thymine, GCTA. Ribonucleic acids lack thymine, and instead uracil is in thymine's place. So CGAU. Additionally, there is some structural differences between the two. DNA typically exists in a double-stranded form. We're most familiar with this with our genome is stored as double-stranded DNA, and RNA is generally a single-stranded molecule. And we'll cover a little bit more in detail what we mean by these strands exactly. So we're going to work on a polymerization here with these two monomers. So we have two nucleotides, and they're going to combine together. Here we have highlighted a hydrogen and a hydroxyl group. So you guessed it, we're going to have a dehydration synthesis. Okay. And that 3' carbon's oxygen is going to bind to the 5' carbon's phosphate. So in a sense, you can see that these are bound between the 3' oxygen of one ribose and then the 5' phosphate of another ribose. And that is how it is polymerized. And we can do this on and on in both directions until we get a DNA strand. Now, the strand, the backbone, as you'll see here, the red backbone, not the rungs if you think of it as a ladder, right? The red atoms you're seeing there, what we're talking about here is the phosphate and the ribose, which make the sides of the ladder. Okay, that's called the backbone. The rungs that go through the middle, those are the nitrogenous bases that interact with each other. And those rungs are where the information is stored. So nucleic acids store and they can transmit genetic information, and the sequence of those nucleotides spell out instructions for the structure and function of an organism. Checkpoint 4. Is the nucleotide below found in DNA or RNA? Name two traits that tell you this. And finally, our last macromolecule are proteins. Now, proteins are made up of the monomers called amino acids. Amino acids have a central carbon atom bonded to four things. They're bonded to an amino group and to a carboxylic acid group. Additionally, there's a hydrogen atom. And what's unique for each type of amino acid is what we call the R group or the variant group. Here we have glycine. Glycine is the most basic amino acid. Its R group is simply another hydrogen. Here's an example of another amino acid, same structure. The central carbon, the amine group, the hydrogen atop, the carboxylic acid to the right, and below the R group or variant group is another carboxylic acid attached to a CH2 group. That's aspartic acid. You don't have to memorize aspartic acid. You should be aware of glycine since it's the simplest. Here's another example, serine, which has a hydrocarbon and then a hydroxyl group for its R group. Now, these are all of the amino acids that you will find in life here on Earth. We can categorize them based upon different features. So the top row here are nonpolar amino acids. So their R groups are nonpolar, all of those. The second row are polar amino acids. So as you can see, serine, it has that hydroxyl group. We discussed the differences in the electronegativities of oxygen and hydrogen. And so we can tell that that's going to be a polar bond there on that R group. And additionally, we can look at the electrically charged groups. So for the acidic groups, we have carboxylic acids that end on those R chains. And for basic groups, remember base is kind of the opposite of an acid. Basic groups have that NH3 plus or an NH2 plus or an NH plus. Those are your different basic groups. And if you just take a brief glance at many of these amino acids, you're going to recognize some of them. Glycine, you've probably heard before. We've also heard quite a bit from alanine when it comes to nutritional supplements and isoleucine. Tryptophan, some of you might have heard the story that tryptophan in turkey is what causes you to be tired. There's not a lot of good evidence that that's the case, but tryptophan you might be familiar with. If you look on that second row there, you might be familiar with glutamine. Glutamine, the polar amino acid. If we've heard of monosodium glutamine, that is sodium glutamine. It's a salt that simply just has glutamine attached to it. And some of you may have heard of some of these electrically charged amino acids as well. So, checkpoint 5, which amino acid would be able to dissolve in water? Asparagine or tryptophan, and why? Okay, now let's study how we get from the monomers to the polymers. You're going to be seeing a pattern here. If you can just look at these two amino acids, you might be able to pick out how they're going to combine together. And that's right, we're going to be doing another dehydration synthesis reaction. We have the hydroxyl group of the carboxylic acid and the hydrogen of the amino group. Off comes our water. The carbon now is free to interact with the nitrogen. And here we get a nice little polymer of two different amino acids. So this gets a little bit complicated when we extend it out here, keeping track of which carbon is which. But if you look carefully, you can pick out the central carbons here. Um, the carbon that is double bonded to the oxygen, that's our carboxylic acid. And we can see it's bound to a nitrogen. So that was our bond between two different monomers. And so then we just look one step further in either direction, and we're going to find our central carbon, okay, and have our orientation there. And in this nice little picture here, it's drawn it so that the variant groups are always facing downwards. Um, but the orientation can change quite a bit, which we'll see later. So it's neatly drawn out here. We can see the glycine in the very first, just that hydrogen group, and different other side chains for different amino acids. So you can imagine that proteins, with all of their interesting functional groups, you can get some really interesting properties that start to emerge with proteins. This example here is going to show you how life takes these amino acid chains and really runs with it to create really interesting structures. So at the very top, we have our basic amino acid chain with the different amino acids represented as circles. They're bound together with those little dashes in between. And down below, you can see that you can take that chain and you can form a pleated sheet with it, or an alpha helix, or different structures. This is done with the different types of amino acids that are being used in this chain here to get those different structures. And then you can take it even further, and you can have the maybe pleated sheets interacting with alpha helixes, et cetera, and fold back on itself to make an even more complex structure. And that's why proteins, you can look at a protein and it can look so messy, is because there's so much going on here with the complexity, and not just the order of amino acids at the very top, but how those amino acids bend and twist to form different shapes, and then how they fold back on themselves to make even more complex hierarchies. So proteins are incredibly versatile and extremely important. In fact, before we understood that DNA was the storage of information in the cell, a lot of scientists had hypothesized that it was going to be proteins that were the storage function cell because of all of the versatility. One of their main roles that they're going to play is structural roles. So, for example, collagen that composes your tendons, the keratin that composes your hair. Additionally, we'll find that it might have different functions such as hemoglobin, which carries the oxygen through your bloodstream. In the bottom right, we see an example of hemoglobin. And then catalytic as well. So most enzymes are going to be proteins. Um, and what catalysis means is that it allows reactions to occur more easily with less energy. In the next slide, we'll watch a short video that demonstrates how proteins fold into complex three-dimensional structures.
Proteins are building blocks that help give your body structure and do work around your body. They move molecules, they make new molecules, they recycle old molecules, and this is just to name a few of the things that they do. But maybe the most interesting thing is that these protein building blocks are themselves made up of smaller building blocks called amino acids. Here you can see a string of amino acids. The different shapes that you can see represent the atoms that make up each amino acid. And here we've highlighted 12 individual amino acids. To make it simpler, each different amino acid can be represented by a single letter. Now each amino acid is shown as a colored ball, looking like beads on a string. This makes the protein structure easier to imagine. The order of amino acids is only part of the story. Because of the different shapes of the individual amino acids, they like to fold into even more interesting three-dimensional shapes. This molecule is twisting into several different spiral or helical shapes, and then those are folding on each other. Take a look at the three-dimensional shape as we give the protein a spin. Kind of looks like logs stacked in a fireplace. Here is the one-letter amino acid code revealing the identity of each amino acid. Again, now you see the amino acids drawn to show the position of each atom. This is like looking at an atomic skeleton of each amino acid. Just like you take up more space than just your skeleton would, see how much space each atom really occupies. This is the real shape of the protein.
Now that we have the basic idea, the primary structure of a protein is the amino acid chain before any folding has been established in the protein. Sometimes this is simply written down as letters representing each amino acid in the chain, and sometimes visually depicted as a sort of loose chain-like structure as shown here. The only interactions we're representing here are the covalent bonds between the amino functional group of one amino acid and its neighboring amino acid's carboxyl acid group. Now, secondary protein structures are very basic. The secondary structure of a protein forms when hydrogen bonds between amino acids cause the chain to fold into specific patterns. Here we can see the specific patterns taking place here. So we have what's called a beta pleated sheet. That's not necessary to memorize beta or alpha sheets, but this now secondary protein structures are very simple in nature. The secondary structure of a protein forms when hydrogen bonds between amino acids cause the chain to fold into specific patterns. The two specific patterns we're showing here are pleated sheets, which kind of look, like a sheet of paper that's been folded up into a pleated pattern, and an alpha helix, which has that helical structure to it. This image here shows how these are formed by looking at the atoms of those amino acids themselves. So if we look at the pleated sheet first, we can see on the very far left of that image, we see the nitrogen's hydrogen of that amino group is interacting with the carbon's oxygen in what used to be that carboxyl group before the amino carboxyl bond was formed there. And that hydrogen and oxygen form that hydrogen bond and pull the chain in on itself in order to form more of a sheeted piece of amino acid structure or protein structure instead of just that loose chain structure we saw in the primary protein structure. The helix shown on the right side is very similar. We again just have some hydrogen bonding occurring between the carboxyl and the amino group of different amino acids. And what determines if a amino acid chain is going to make a pleated sheet or a helix really comes down to the amino acids that are in that specific chain so that they can spatially arrange with one another in order to make those hydrogen bonds occur. And then lastly, we have the tertiary protein structure. The tertiary structure is when the secondary structures fold and interact with each other. And this is caused by hydrophobic effects on the R groups and also their electrostatic interactions with one another. So this image is going to depict the hydrophobic interactions that we see. So on the far left, we have just a primary structure of an amino acid, or it could be maybe even a secondary here. The relationship is still going to be the same. You're going to have those side chains, those R groups. Some of them are going to be polar side chains. Some of them are going to be non-polar side chains. Now, the polar side chains are more than happy to interact in water, aqueous solutions. Remember, whenever we're talking about the inside of cells, they're going to be it's an aqueous solution in there. It's a water-like suspension inside of a cell, and that's going to be the case outside of the cell as well. So these proteins are going to auto-assimilate in a way that has those polar R groups sticking outward and the non-polar side chain sticking inward, and that'll lead to this hydrophobic effect of the way the proteins fold. So it doesn't take any sort of intelligent interactions here to get this done. It's just natural chemical reactions, which forces this protein into a very complex shape. All right. Now, in this image, we have some of the amino acids that have electrical charges with one another. And these R groups, if you have a negative charge and a positively charged amino acid, they're going to be attracted to each other. And that's what we mean by these electrostatic interactions, where if they're oppositely charged, they're going to be brought together. If they're negatively charged, they're going to repel each other. Checkpoint 11. Name a pair of amino acids whose R groups would be electrically attracted to each other. Okay. So, and now finally we have the sulfide bridges. So, in cysteine, they have a sulfur group in the amino acid. And what's unique about this sulfur group is the sulfurs can interact with another cysteine to form a sulfide, what we call a sulfide bridge. Okay. I'm going to show how that looks here. So here we have some hydrophobic interactions. We have some hydrogen bonding occurring between amino acids. And additionally, we have two cysteines right there in the middle. And when those two sulfur hydrogen chemicals bonds get in close proximity to one another, this one sulfur can react with the other sulfur and form these pretty strong disulfide bridge connections, which hold the amino acid chain together there. And we see this in our daily lives. We have keratin, the main protein found in hair. It contains cysteine amino acids in pretty high amounts. And this leads to the formation of disulfide bridges with each other. And this actually is what causes hair to become curly. You can apply heat to break the bond between those sulfur atoms, and this causes curly hair to become more straight. All right. And the final step is what we call the quaternary protein structure. And this is when you have more than one amino acid chain that clumps together or interacts together and creates a multi unit structure. This does not happen with all proteins, but many proteins have different what we call subunits. So here's a great example of that. Here we have hemoglobin, which is made up of four separate amino acid chains which clump together. And as you know, hemoglobin is responsible for carrying oxygen around in our blood, and it has an iron or a heme complex in the middle of each of those subunits. There's a great game out there if anyone wants to try it out. It's called Fold It. Fold It is a protein puzzle game that allows you to try and figure out how to fold proteins. And you can. This is a bit of a citizen science activity here. Proteins are a little bit difficult to figure out how they fold, and we can use their folding in order to try and solve problems. So, trying to make proteins that might be good antivirals against the coronavirus. Okay, checkpoint 7. I think I misspoke and said checkpoint 11 before. That was checkpoint 6. This is checkpoint 7. Match the following biological macromolecules with the monomers that compose them. Point 8. Match the following biological macromolecules with their function in biological organisms. Let's return to the Miller-Urey experiments and see what their results were. Checkpoint 9. One of the products of the Miller-Urey experiments was the molecule on the left. Which macromolecules could be formed from this molecule? Checkpoint 10. Later similar experiments also discovered the molecules on the left. Which molecules could be formed from this molecule? And checkpoint 11, identify the following biological molecule and the indicated functional group. Finally, checkpoint 12, identify the following biological molecules and the indicated functional group. And that wraps up chapter 3. Thank you so much for joining, and we'll see you on the next one.